Okay, welcome back, y'all. And this is what I wanted to show you about the Kaplan books. So, I let's first things first get the font a bit smaller. So we have 2 pages. Now, here, this is what the book is about. It's like 1,000 pages for the biology section, and also something cool for the I think it's about the MCAT here. We definitely want to get the structure down, know everything. So we have to know every detail about the exam. So we should know that the first section, physics and chemistry, is 95 minutes. It's 59 questions, and 44 of them are passage questions. And the rest, which is 59 minus 44, it's 15. So 15 questions are standalone questions, like discrete questions. And it's true for the biology section and the psychology section as well. So it's only cars that is 90 minutes and 53 questions. And you can see here. So this is just what I said. And this is what caught my attention actually. So I don't know why this image is too low of resolution, but yeah, you can see here the dark blue is actually biology, and you can see that for the biological and biochemical section, it consists of more than 60% of the content. Even there is a bit of like 5% in the physics and chemistry, and there's still a bit even more, a tad more like in the psychology section. And there's also the MCAT. In total, MCAT, we have over 20, so like 25% of the whole section is in biology. And you can see that psychology is a bit less. It's the second rank here, and then we have the biochemistry. So biology and biochemistry together consist 40% of the exam, which is crazy. And we have, I think, after psychology and biochemistry, we have the general chemistry, and same, like a tad less, it's sociology, and then we have the physics, and last but not least, we have organic chemistry. But regardless, we have to study the shit out of all these sections. However, our major focus, we should know the ins and outs of the biology section and the biochemistry section, right? And that's why the biology book here, these 1,000 pages are super important. And let's look at this actually here. So let's read this quickly. Is there anything before this about Scientific American? The preface. I like reading these a lot actually. So, and these are, yeah, this is the title. So, I mean, I'm reading from the book, and I've seen others reading from books before. So I hope this is not a copyright issue. And if there is any, I would be more than willing to take this down, and but like share it in a private setting if you all need it. So let's go with this. So So the AMC has defined 4 scientific inquiry and reasoning skills, SIRS, that will be tested in the 3 science sections. We have 35% of questions asking for the knowledge of scientific concepts and principles. So that is straight from our knowledge pool, our cookie jar, whatever you want to call it. Then 45% of questions, more than that even, we have scientific reasoning and problem solving. So you want to be able to apply these principles and concepts to reasoning and problem solving. And then we have 10% of the questions on reasoning about the design and execution of research, and another 10% on database and statistical reasoning. Let's see how each one breaks down into more specific test day behaviors. Note that the bullet points of specific objectives for each of these series are then directly from the official guide of the MCAT exam. The descriptions of what these behaviors mean and sample question stems, however, are written by Kaplan. Knowledge of scientific concepts and principles. This is probably the least surprising of the 4 series. The testing of science knowledge is, after all, one of the signature qualities of the MCAT. Skill 1 questions will require you to do the following: recognize correct scientific principles, identify the relationships among closely related concepts, identify the relationships between different representations of concepts, verbal, symbolic, graphic, identify examples of observations that illustrate scientific principles, use mathematical equations to solve problems. At Kaplan, we simply call these science knowledge or skill 1 questions. Another way to think of skill 1 questions is as one-step problems. The single step is either to realize which scientific concept the question stem is suggesting, or to take the concept stated in the question stem and identify which answer choice is an accurate application of it. Skill 1 questions are particularly prominent among discrete questions, those not associated with a passage. These questions are an opportunity to gain quick points on test day. If you know the science concept attached to the question, then that's it. On test day, 35% of the questions in each science section will be skill 1 questions. Here are some sample skill 1 question stems. How would a proponent of the James Long theory of emotion interpret the findings of this study cited in the passage? So just, you need to know about the James Long theory of emotion. Which of the following most accurately describes the function of FSH in the human female menstrual cycle? So which of the following most accurately describes the function of FSH in human female menstrual cycle? You just need to know the knowledge. If the products of reaction 1 and reaction 2 were combined in solution, the resulting reaction would form. Ionic bonds are maintained by which of the following forces? Then we have skill 2, so scientific reasoning and problems. Let's look at some examples. Reason about scientific principles, theories, and models. Analyze and evaluate scientific explanations and predictions. Evaluate arguments about causes and consequences, causes and effects. Bring together theory, observations, and evidence to draw conclusions. Recognize scientific findings that challenge or invalidate a scientific theory or model. Determine and use scientific formulas to solve problems. Just as skill 1 questions can be thought of as one-step problems, many skill 2 questions are two-step problems. In more difficult skill 2 questions may require 3 or more steps. These questions can require a wide spectrum of reasoning skills, including integration of multiple facts from a passage, combination of multiple science content areas, and prediction of an experiment's results. Skill 2 questions also tend to ask about science content without actually mentioning it by name. For example, a question might describe the results of one experiment and ask you to predict the results of a second experiment without actually telling you what underlying scientific principles are at work. Part of the question's difficulty will be figuring out which principles to apply in order to get the correct answer. On test day, the majority, like 45% of the questions in each science section, will be skill 2 questions. Here are some examples. Which of the following experimental conditions would most likely yield results similar to those in figure 2? So we have to apply the experimental conditions yielding results similar to figure 2. So we have to look at figure 2, know the ins and outs of it, and then apply it with the experimental conditions. All of the following conclusions are supported by the information in the passage except, so we have to know how every option, the conclusions, are supported by the information except one of them. The most likely cause of the anomalous results found by the experimenter is, so which of the options is most likely the cause of the anomalous results found by the experimenter, and impact to a man's chest. An impact, I like to highlight it green. Green for me implies the fact that I have a takeaway from this. So these are examples which are good takeaways. An impact to a man's chest quickly reduces the volume of one of his lungs to 70% of its initial value while not allowing any air to escape from the man's mouth. By what percentage is the force of outward air pressure increased on a 2 cubic centimeter portion of the inner surface of the compressed lung? Skill 3, reasoning about the design and execution of research. The MCAT is interested in your ability to so remember we have 4 skills. To critically appraise and analyze research. Identify the role of theory, past findings, and observations in scientific questioning. Identify testable research questions and hypotheses. Distinguish between samples and populations and distinguish results that support generalizations about populations. Identify independent and dependent variables. This is a good one. There is the Shematian website, consulting website, and they have really good, in my opinion, methods and summarizations of each section. So we definitely want to go over those as well. And it's about independent. They have a TADE-P model method, which is the title, the axes, and the independent and dependent variable. The x-axis is, I believe, the independent variable, and the y-axis is the dependent one. So the independent variables, you can change them, but the dependent variable depends on those independent variables, right? So on the x-axis, you usually have, for example, let's say you have the time. Usually that's the case, right? I mean, it's a common one in general world. So, for example, the time is something that you can control. It's an independent variable. But whatever you are measuring to be affected by time is a dependent variable. For example, the size of the molecules are changing through days of the experiment that you're doing. So the size is the dependent variable. Then we have reason about the features of research studies that suggest associations between variables or causal relationships between them, such as temporality and random assignment. Identifying conclusions that are supported by research results. determining the implications of results for real-world situations, and reasoning about ethical issues in scientific research. So medical schools over the years are requiring more practical research skills of MCAT test takers, and skill 3 questions are the response to these demands. So this skill is unique in that the outside knowledge you need to answer skill 3 questions is not taught in any one undergraduate course. Instead, the research design principles needed to answer these questions are learned gradually throughout your science classes, and especially through any laboratory work you have completed. It should be noted that Skill 3 comprises 10% of the questions in each science section on test day. Examples: what is the dependent variable in the study described in the passage? The major flaw in the method used to measure disease susceptibility in experiment 1 is. So we have dependent variable, we have a major flaw in the method. Which of the following procedures is most important for experimenters to follow in order for their study to maintain a proper randomized sample of research subjects? A researcher would like to test the hypothesis that individuals who move to an urban area during adulthood are more likely to own a car than are those who have lived in an urban area since birth. Which of the following studies would best test this hypothesis? Then we have last but not skill, which but not man, my wording. So we have skill 4, database and statistical reasoning, and the science section tests your ability to analyze visual and numerical results of experiments in studies, use, analyze, and interpret data in figures, graphs, and tables, evaluate whether representations make sense for particular scientific observations and data, use measures of central tendency, mean, median, and mode, and measures of dispersion, range, interquartile range, and standard deviation to describe data, reason about random and systematic error, reason about statistical significance and uncertainty, interpreting statistical significance levels and interpreting a confidence interval, use data to explain relationships between variables or make predictions, use data to answer research questions and draw conclusions. Skill 4 is included in the MCAT because physicians and researchers spend much of their time examining the results of their own studies and the studies of others. So it's very important for them to make legitimate conclusions and sound judgments based on the data. According to the information in the passage, there is an inverse correlation between types of errors is most prevalent in this test. If a fourth trial of experiment 1 was run and yielded a result of 54% compliance, which of the following would be true? If this was true, then which of the following would be true? So, science knowledge, skill 1, do you remember this science content? Skill 2, critical thinking. Do you remember this science content? And if you do, could you please apply it to this novel situation? Could you answer this question that cleverly combines multiple content areas at the same time? And then skill 3, experimental and research design. Let's forget about the science content for a while. Could you give some insight into the experimental or research methods involved in this situation? And skill 4, let's forget about the science content for a while. Could you accurately read some graphs and tables for a moment? Could you make some conclusions or extrapolations based on the information presented? Okay, cool. Let's now, we have cars. So foundations of comprehension is the main idea. Detail, scattered detail according to the passage, which of the following is false about literary reviews in the 1920s. Inference, implication, inference, assumption. What is the difference between implication? So implication, you want to imply, like, which of the following phrases as used in the passage is most suggestive that the author has a personal bias toward narrative records of history. So which of the following options implies that the author has a personal bias? But assumption is in putting together her argument in the passage, the author most likely assumes. So what is the author assuming in the passage? So the first one is that implications are types of inferences that suggest. So like if any of these following phrases, like any of them that are used in the passage, which of them is most suggestive that the author has this idea, right? Has this method or thinks what the author thinks about. But the assumption is putting the argument in the passage that the author is most likely to assume about the passage, right? So the second one is like, what does the author assume? Assumptions are, what does the author assume? And implications are, which of the phrases imply that the author thinks like this or has this behavior. Definition in context, the word obscure, when used in reference to the historian's actions, most nearly means. So in the context of the passage, what was the definition of this? So this was for foundations of comprehension. I apologize for that. I hope you are not listening to my voice with maximum volume on your headphones, and you might have gone deaf after this. Okay, we do have reasoning within text. So while foundations of comprehension questions will usually depend on interpreting a single piece of information in the passage or understanding the passage as a whole, reasoning within text requires more thought because they will ask you to identify the purpose of a particular piece of information in the context of the passage, or ask how one piece of information relates to another. Reasoning within text will require you integrate different components of the text to draw relevant conclusions. It will ask you to judge certain parts of the passage or even judge the author. This will fall under the reasoning within text. It can ask you to identify authorial bias, evaluate the credibility of cited sources. I know that we are supposed to do biology for this hour, so that's why I want to just go over this very quickly. So we have the function, but for now we can just highlight these, right? And so for this, we have reasoning within text and goes here. Judge certain parts of the passage, or even judge the author. Function, the author's discussion of the effect of socioeconomic status on social mobility primarily serves which of the following functions? Strength and weaken. Which of the following facts is used in the passage as the most prominent piece of evidence in favor of the author's conclusions? Based on the role it plays in the author's argument, the possessed can be considered. And then we have reasoning beyond text. Beyond, so extrapolate ideas from the passage to new concepts, or assess the impact of introducing new factors to ideas from the passage. So either you have to apply the ideas from the passage to a new situation, or you have new information, or conditions to ideas from the passage. So you want to assess the impact this new information has on the ideas from the passage. So examples are: apply if a document were located that demonstrated Berlioz intended to include a chorus of at least 700. Okay. Cool. So what I actually like about these, let's go over these. Okay, is the testing. So let's do these actually, and then we can see this should be also a review for us on chapter 1. Hyperbaric oxygen, and then based on the questions we answer, like the 15 questions, we can decide how much time we want to spend on the passage. But still, this is assessment questions and very discrete type questions, so we should actually do them. Hyperbaric oxygen may be used as a treatment for certain types of bacterial infections. In this therapy, the patient is placed in a chamber in which the partial pressure of oxygen is significantly increased. So increase the partial pressure of oxygen. Increasing the partial pressure of oxygen in the patient's tissues. This treatment is most likely used for infections with obligate aerobic, facultative anaerobic, aerotolerant anaerobic, obligate anaerobic bacteria. Perfect. So we have an oxygen situation. So the patient is placed in a chamber in which so you increase the partial, and this increase of oxygen is used as a treatment for certain types of bacterial infections, right? So these bacterial infections will be killed, right? So these bacteria will be killed. This treatment is most likely used for infections because these bacteria are killed, it means that they don't like oxygen, right? So they should be obligate anaerobic bacteria. So it's D. Let's put our answers. Let's actually put it here. We have chapter 1 for here. So let's do Kaplan assessments cues. We have 1D and then we can do the rest. Yep. Okay. Which of the following does not describe connective tissue cells? They account for most cells in muscles, bones, and tendons. That is correct. So it does describe connective tissue cells. They secrete substances to form the extracellular matrix. Muscle, bones, tendons. Do they secrete? For now, in organs, they tend to form the stroma. So between B and C, I would choose C. In organs, they provide support for epithelial cells. So we have endothelial cells that are in capillaries, and epithelial cells are in digestive system, lungs, right? And these epithelial cells in organs, they provide support for them. Sure. So they tend to form the stroma. Stroma. Where have we heard this before? Is it for neurons? They secrete substance to form the extracellular matrix. Yeah, I mean, C should be the incorrect one, which is the answer for here. So I would go with C. Which of the following types of nucleic acid could form the genome of a virus? So we basically want to see which one could not be right. Single-stranded RNA, double-stranded DNA, single-stranded DNA. So we can have both 1 and 3. Double-stranded DNA, we know it's a thing for us humans, but even viruses have it. So I would go with all of them. As far as I know, I saw all of like we have the single-stranded or double-stranded factor, and there's also the RNA or DNA factor. So it should be there. The theory of spontaneous generation states that living organisms can arise from non-living material. In 1859, Pasteur demonstrated that no organisms emerged from sterilized growth media, weakening the theory of spontaneous generation and supporting which tenet of cell theory. So, spontaneous generation, living organisms can arise from non-living material. Pasteur demonstrated that no organisms emerge from sterilized growth media, weakening the theory and supporting which tenet of cell. All living things are composed of cells. The cell is the basic functional unit of life. Cells arise only from pre-existing cells. Cells carry genetic information in the form of DNA. I mean, I'm not sure if this is part of a cell theory, but it seems like the best answer suiting for the fact that cells arise only from pre-existing cells. However, we have also the cell is the basic functional unit of life. So all living things are composed of cells. That's also do we have any other living things that are not composed of cells? Or, for example, bacteria and yeast considered as cells? You know, we have prokaryotic and eukaryotic cells. So those are just prokaryotic. And living organism can arise from non-living material, emerge from sterilized growth media. So I would say, yeah, I would say C, but this is a tricky one. C, B, like in order I would go C, and then A, and then B, But yeah, C should be like B, I would say. C, B, and then A. Mitochondrial DNA is circular. I think so. I think we read it and it was circular. Self-replicating. Single-stranded. I think it's 1 and 3, which we don't have it. So self-replicating. We know mitochondrial DNA comes from our mothers. So 1 and 2, single-stranded. 1, 2, 3, single-stranded. If it's a DNA, it's a double-stranded. I mean that deoxyribonucleic acid. Okay, we could have single-stranded. Let's go with all of them. Which of the following is not a function of the smooth endoplasmic reticulum? We know smooth SER is for lipid synthesis, yes. Poison detoxification, it does that too, surprisingly. Protein synthesis, hell no. Transport of proteins, sure, but protein synthesis, that happens in rough ER, or ribosomes, stuff like that. Yeah, I think it's C. What is the main function of the nucleolus? So nucleolus is for making the ribosomes, ribosomal RNA synthesis, right? DNA replication, cell division, chromosome assembly. So which one is the main function of nucleolus? I think replication, DNA replication, happens in nucleolus. We don't want to replicate it outside of the cell. It might be harmed, right? So I believe it's B. Which of the following organelles is surrounded by a single membrane? Single membrane. Mitochondria, no. Lysosomes, no, I don't think so. They need to be detoxifying. So nuclei, no also. And ribosomes, I would go with ribosomes. Which of the following is not a difference that will allow one to distinguish a prokaryotic and a eukaryotic cell? So which of them is not a difference between prokaryotic and eukaryotic cells? Ribosomal subunit weight. So it should be like a similarity between prokaryote. Man, might be even A. Presence of a nucleus. Yeah, there is a difference between them, so not B. Presence of a membrane and on the outside surface of the cell. That's also on the outside of the cell. That's for prokaryotes, so that's also a distinguishing factor. Presence of membrane-bound organelles. That's also yeah, it should be A. I mean, still the ribosomal subunit weight is different between prokaryotic and eukaryotic cells. So presence of a nucleus, presence of a membrane on the outside surface of the cell. Membrane-bound organelles, we know they are for eukaryotes. And presence of a nucleus is, but a membrane on the outside surface of the cell. I think it's C then A. So I would go with C. Ribosomal subunit, presence of a nucleus. They don't have a membrane-bound nucleus. but they do have nucleus, right? I'll go with B. I'm probably doing the wrong thing, changing my answer. But anyway, which of the following does not contain tubulin? So, it's the hard stuff like cilia, flagella, microfilaments. Those are actins, not tubulins. And centrioles have tubulins, so it's microfilaments. 10 is C. Herpes, HSV, enters the human body and remains dormant in the nervous system until it produces an outbreak after exposure to heat, radiation, or other stimuli. Which of the following statements correctly describes herpes simplex virus? While it remains dormant in the nervous system, this virus is in its lytic cycle. So we have lysogenic and lytic cycle. So lytic is when so it's dormant until it produces an outbreak. So during an outbreak, the virus is in the lysogenic cycle. So what is the difference between lytic and lysogenic? In lytic, it results in cell death, so it kills the cell. So it's definitely not that for dormant. Let's see C and D. So it after the outbreak is lytic cycle, I believe. Herpes and lysogenic is just leaving the cell, like being dormant until there's an outbreak. I think it's B. Herpes simplex virus adds its genetic information to genetic information of the cell. The herpes simplex virus contains a tail sheath and tail fibers. So it's dormant. The tail sheath thing is for bacteriophages. So honestly, during the outbreak, until there is an outbreak after exposure to heat, radiation, or other stimuli. I think it's B. Okay, I think I'm writing somewhere else. Just right here, man. Oh, my bad. Okay. 12, right? Resistance to antibiotics is a well-recognized medical problem, which mechanism can account for a bacterium's ability to increase its genetic variability and thus adapt itself to resist different antibiotics. So we know antibiotic resistance is okay. So it gives it superpower. So binary fission is for bacteria, completely bullshit. It's like a replication thing, so it's not for which mechanism can account for ability to increase genetic variability and thus adapt itself to resist different antibiotics. This is about the transduction thing, right? Where it is the thing is injected, goes into the DNA of the plasmid can go to. So plasmid we had a transduction and transformation. So I had associated the P of plasmid to something, but somehow it doesn't make sense to any of them. So we need to refine that because conjugation, plasmid, like transduction, transformation, all of them have P. Plasmid has D, but it wasn't the D, it was the P. So the conjugation was also part of it. I think it was like it's 2 and 3, but we need to review it. A bacterial cell is noted to be resistant to penicillin. The bacterium is transferred to a colony that lacks the fertility factor, and the rest of the colony does not become resistant to penicillin. However, the penicillium resistant cell has also started to exhibit other phenotypic characters, including a secretion of novel protein, which of the following methods of bacterial recombination is not likely to account for this change. I read it too fast, so let's do it again. A bacterial cell is known to be resistant, so it's resistant to penicillin. The bacterium is transferred to a colony that lacks the fertility factor, and the rest of the colony does not become resistant to penicillin. However, the penicillin resistant cell this is a Griffith experiment, no? Has also started to exhibit other phenotypic characteristics, including secretion of a novel protein. Which of the following methods of bacterial recombination is not likely to account for this change? So we have conjugation, we have transformation, infection with a bacteriophage, transduction. I would say infection with a bacteriophage. Man, these questions are not easy for me. I did really easy well on the first chapter of organic chemistry, but not these. In Alzheimer's disease, a protein called the amyloid precursor protein is cleaved to form a protein called beta amyloid. So this protein is cleaved to form a protein called beta amyloid. This protein has a beta pleated sheet structure and precipitates to form plaques in the brain. Oh, this is like the whatever the name. I mean, these names of these scientists are super hard for me to keep in mind, but definitely need to review that. This mechanism of disease is most similar to which of the following pathogens? But this was a special case of these prions that are protein infections, right? We know the viroids are plant pathogens. Oh, it was a plant pathogen, but they witnessed this in the brain too, but it was the prions. The prions are the ones that do the beta pleated sheet thing. So let's go with C for 14. And last one. After infection of a cell, a viral particle must transport itself to the nucleus in order to produce viral proteins. So the viral particle must transport itself to the nucleus in order to produce viral. What is likely the genomic content of the virus? Ooh. So if it has to go to the nucleus in order to produce viral proteins, it is done probably double-stranded DNA. And then we have positive sense or negative sense. Okay, these actually gave me a good hard time, but maybe double-stranded RNA. Man, these are hard. Not easy. Okay. Answer key 1 is D. So how do we, let's see if this would be a good idea. 1 is D. Then we have 2 as A. So yes, we're getting the incorrect ones. 2, and then 3 is D. That is correct. 4 is C. 4 is C. We have apparently chosen the right answer. 5 is C. That is wrong. I'm definitely going to get more, like a 4 or 5 wrong at least. 6 is C. Yes. And then 7 is A. But got it wrong. 8 is A, 9 is C, 9 is C, and then we have 4 C's till 12. So now, yes, no, and yes, and we have ACA. ACA, wrong, correct. Okay. Now we can review and learn, right? Man, we have even more wrongs than correct ones. What a shame. Honestly, this is not like the cell is not my forte at all. But we're here to learn about it. So, nice thing is that we have the explanations on at the end of the chapter, the concept summary. Like it's at the very end, I believe. Let's see. We have concept summaries here. Yes, it's here. So first one, we got it right. Obligate anaerobes cannot survive in the presence of oxygen and would like to be killed. In such a therapy, treating the infection, the other types of bacteria listed can all survive in the presence of oxygen. So infections involving these. Perfect. We have the obligate anaerobe stuff under control for now. 2. We chose C, which is wrong. While bones and tendons are composed predominantly of connective tissue cells, muscle tissue is considered a different tissue type. Other examples of connective tissue include cartilage, ligaments, adipose tissue, and blood. So muscle tissue is considered a different tissue type. Connective tissue often secretes substances to form the extracellular matrix, such as collagen and elastin. Eliminating B, C, and D are, so we chose C. C and D are essentially identical and can both be eliminated. In organs, connective tissue often forms the support structure for epithelial cells called the stroma. So A is the answer. Now, do we just go back here? What do we do? So 2 is A. They account for most cells in muscles, bones, and tendons. Oh, muscles are different. What? So they are not part of the connective tissues. Okay, our timer is saying that we should start the psychology and sociology section. We didn't even get to cover chapter 2 for biology. It's fine, we will come back to it. But let me finish this first, and then we come back to the psychology and sociology section. So connective tissues do secrete extracellular matrix. They do tend to form the stroma, and they do provide support for epithelial cells. What is stroma? Man, I need my chat GPT here. Okay, I'm somehow connected to this, which we don't want right now. What is stroma? Refers to colorless fluid surrounding the grana within the chloroplast, within the stroma. Okay, it would be actually good to, so we have these 4 sections, right? And everything that we learn as we go down, we build on it. But I want missed questions, right? I mean, it's still good to have missed question under these, right? So we can actually write under this. And we can go with this format. Why I missed it? So we have biology, chapter 1, CH1, and SSQ, Kaplan. So we have Kaplan, SSQ 2. Why I missed it is I didn't know what stroma is. What is stroma? So stroma are in the supportive tissue of an epithelial organ, tumor, gonad, consists of connective tissues, and blood vessels. Refers to colorless fluid surrounding the grana within the chloroplast. Within the stroma are grana, the suborganelles where photosynthesis is started before chemical changes. Fluid filling up the inner space of the chloroplast. It's a fluid. So why I missed it? What I learned. Let's actually zoom in on this. What I learned is that muscle is not connective tissue, but tendon and bones are. What else? Connective tissue secrete substances to make extracellular matrix. extracellular matrix, and they also form stroma and provide support for epithelial cells. Okay. The explanation again, while having in mind that which of them could form a genome of a virus. So we go again to was it it's better to go to the end there. Bones and tendons are composed predominantly of connective tissues. Muscle tissue is considered a different tissue type. Other examples of connective tissue: cartilage, ligaments, adipose tissue, and blood. Tendons, bones, blood. So blood, tendons, and cartilage. Tendons, cartilage, ligaments, bones, blood, adipose, okay. Oh, so in organs, connective tissue often forms the support structure for epithelial cells, called the stroma. So in organs, connective tissue provide support for epithelial cells, which is called stroma. It gives structure. So 3 we got right. We chose D. It's all of them, right? Single-stranded, double-stranded, either DNA or RNA. Can be either single. Therefore, all the types of nucleic acids listed here could be useful for a viral genome, making D the correct answer. C is for the process of sterilization kills all living cells. A lack of cellular growth in these conditions supports the idea that cells can only arise from pre-existing cells. By contrast, since the experiment did not directly visualize the cells, nor did it analyze the genetic material, A, B, and D can be eliminated. Now, why did we get 5 incorrect? Mitochondria are thought to have evolved from an anaerobic prokaryote, engulfing an aerobic prokaryote and establishing a symbiotic relationship. Therefore, so it was an anaerobic prokaryote engulfing an aerobic one, which established a symbiotic relationship. Therefore, mitochondrial DNA, or mDNA, is likely to be similar to bacterial DNA. Both mDNA and bacterial DNA are organized into a single circular chromosome of double-stranded DNA that can replicate during binary fission. Therefore, statements 1 and 2 are correct, while statement 3 is incorrect. Let's get back to... So, mitochondrial DNA is circular and it is self-replicating. I wanted to choose 1 and 2, but I chose single-stranded 2. So it's double-stranded DNA, and be Q5 why I missed it, what I didn't know, mitochondrial DNA is from prokaryotes, roots from prokaryotes, and is double-stranded. So circular, self-replicating, being from prokaryotes means that it is self-replicating, binary fission, and circular. Cool. We have question 6 correct. So which of the following is not a function of smooth endoplasm? Yeah, proteins, but they do transport proteins. Hint Q6 hint SCR still does not synthesize proteins, but does transfer them. Transfer or transport? Transport them. We got question 7 wrong. We chose B. What is the main function of the nucleolus? So if B is not the answer, what would be? Chromosome assembly, cell division, ribosomal RNA synthesis. Yeah, man, I think it's the ribosomal RNA synthesis. 7 is A, right? 7 is A. Yeah, the nucleolus, not to be confused with the nucleus, is a dense structure within the nucleus where ribosomal RNA is synthesized. So where is it? Main function of nucleolus. Synthesizing or ribosomal RNA, right? Or RNA. Yes. So follow-up question would be, wait a second. Can I add this here? Yes. Does nucleolus, where's our, okay, here. Let's actually ask it here. Does nucleolus Does nucleolus have any other functions aside from its main function, which is synthesis of rRNA? While the nucleus is dynamic, ribosome assembly, cellular stress response, cellular aging, senescence, regulation of protein localization, regulation of cell cycle. How about what were the options? Like DNA replication. How about DNA replication? How about DNA replication, cell division, chromosomal assembly, cell division, or chromosomal assembly? Cell division, mitosis, or meiosis, or chromosome assembly. In other cellular compartments, DNA replication occurs in the nucleus, but is not a direct function of the nucleus. It's more focused on ribosomal RNA synthesis. DNA replication involves the synthesis of a new DNA strand based on existing template DNA. It takes place in the nucleus. Cell division, mitosis, nucleolus. So for here, DNA replication, nucleus. Then we have cell division. Was it cell division for real? Yeah. I mean, yeah, that's why we didn't choose it. So in particular, during mitosis, it is not directly involved in the process of dividing. Separation and distribution of chromosomes occur in the nucleus during mitosis. So, yeah, it's again nucleus. Cell division, nucleus, and chromosome assembly. It's a complex process involving correct arrangement of DNA and associated proteins. This occurs in the nucleus, but is not directly associated with nucleolus. The nucleus primarily deals with RNA. Yeah. Chromosome assembly, nucleus. It's all nucleus. Cool. Next question is 8. Question 8, we got it wrong. 8 and 9 as well. So we have 8. Why I missed it. Let's see. Which of the following organelles is surrounded by a single membrane? I chose D. Is surrounded by a single membrane. Nuclei. It's either A or C, and I think it's nuclei. It is the nucleolus that has 2 membranes. So that's C, A, 8. Eight is A. Lysosomes are vesicular organelles that digest material. Using hydrolytic enzymes, they are surrounded by a single membrane. Both mitochondria and nuclei are surrounded by double membranes. Ribosomes must not be surrounded by membranes because they are found not only in eukaryotes but also in prokaryotes, which lack any membrane-bound organelles. So, number of membranes for each cell compartment. So nuclei 2 mitochondria, we know it's 2 lysosome is 1 and ribosomes is 0. Why no membranes for ribosomes? Because they are found not only in eukaryotes but also in prokaryotes. Because why? Because they're found in both prokaryotes and ribosomes and eukaryotes. Both prokaryotes. Next question, question 9. We chose B. Some of the main differences between prokaryotes and eukaryotes are that prokaryotes do not have a nucleus, while eukaryotes do, eliminating B. Prokaryotes have, so they don't have nucleus. Ribosomal, so eliminates A. Prokaryotes do not have a membrane-bound organelle, whereas eukaryotes do, eliminating D. The presence of a membrane on the outer surface of the cell could not distinguish a prokaryotic cell from a eukaryotic one because 1 gram-negative bacteria and animal cells share this feature. Both gram-negative bacteria and animal cells share this feature. Thus, C is the correct answer. The distinguishing factors between pro and eukaryotes. So pro. So these are the information that we learned. Pro do not have nucleus. We know we got the question wrong because pro both gram negative, both pro and U have outer membrane on cell membrane, i.e. gram-negative bacteria and human cells. And the rest is just ribosomal. So differing ribosomal subunit weights. And one more thing. Membrane-bound organelles. Pro don't have Okay, now let's go to the next one. We have 10, which we got correct. Tubulin is the primary. Yeah, we know flagella cilia have tubulin, and also the microtubules, centrioles. They organize microtubules into mitotic spindle. But microfilaments are from actin, not tubulin. We have 11, which we got wrong. Viruses can exist in either the lytic or lysogenic cycle. They may even switch between them. During the lytic cycle, the virus's DNA takes control of the host cell's genetic machinery. So the DNA of the virus takes control of the host cell's genetic machinery, manufacturing numerous progeny. In the end, the host cell bursts, lyses, and releases new virions, each capable of infecting other cells in the lysogenic cycle. So in the lytic, the DNA of the virus takes control of the host's DNA, genetic machinery, manufacturing numerous progeny, and then in the end, the host cell bursts, the host cell lyses, bursts, so it results in cell death and releases new virions, each capable of infecting other cells. In the lysogenic cycle, viral DNA is added to the host cell's genome. So the viral DNA adds to the host cell's genome, where it can remain dormant for days or years. And either spontaneously or as a result of environmental circumstances, the provirus can reactivate and enter a lytic cell cycle. Thus, A and B are incorrect because the terms are reversed. Okay. So if we reverse the terms for A and B, they are correct. They become correct. So what happens in the lysogenic cycle? The viral DNA adds to the host cell's genome, where it can remain dormant for days or years. When the circumstances arise, right, like the heat that was discussed in the question stem, the provirus, either spontaneously or as a result of environmental circumstances, the provirus can reactivate and enter a lytic cycle. I think this was for the herpes simplex virus, right? HSV. And then we have D describes features of bacteriophages, which are viruses that infect bacteria, not the human nervous system. Oh, this was about the brain one, right? And so C accurately describes it was HSV, operates during the lysogenic cycle, making it the correct answer. Let's take a look at the question itself. 11. The HSV enters human body and is dormant, which is following outbreak, dormant in the nervous system. So the reason D is wrong is that it is saying it contains a tail sheath, so it's like in tail fibers, which means it's a bacteriophage. So those are for bacteria, not for human cells. Herpes simplex virus adds its genetic information to the genetic information of the cell. Because it's dormant, it adds its genetic information to it. This is a very informative question. So this was question 11. Lysogenic versus lytic cycles. So we chose B, which was 1 and 3. No, sorry. B during outbreak viruses. So while remaining dormant is for lysogenic, and during outbreak the virus is in the lytic cycle. So during the outbreak, so it produces an outbreak, remains dormant in the universe until it produces an outbreak. So the outbreak is the lytic. Reversed lytic is when outbreak, cell lysis bursts, lysogenic is when it's dormant. And then for option D, containing tail sheath fiber or fiber refers bacteriophage, refers viruses for bacteria, not human nervous system. And we also know that now it adds genetic information to genetic information of the cell. So lysogenic uses host cells genetic machinery Am I right? Let's go back to 11. So during the lytic cycle, virus takes control of host cell. Okay, it was lytic. Don't make the mistake again. Lytic uses host cells genetic machinery. In the end is cell death. Produces progeny. Lysogenic is viral DNA integrated, added to host's DNA. This is genome and dormant for days or even years. Now we have question 12, which we got right. So it would be 13 and 15. We're almost done. So 12, bacterial cells reproduce by binary fission, an asexual process in which the progeny is identical to parent. Therefore, binary fission does not increase genetic variability. Conjugation can be described as sexual mating in bacteria. It is the transfer So sexual mating in bacteria's conjugation is the transfer of genetic material between two bacteria that are temporarily joined. Between two bacteria that are temporarily joined. So it's the transfer of genetic material. Honestly, we could add all of these to our flashcards. So still for question 2, hint 12, we need hints. So hint Q12 hint. Did we review the other ones that we? Yeah, for those we didn't need it. So we have question 12 hint, which is this: binary fission asexual replication for bacteria, therefore no genetic variability, conjugation, sexual mating, for bacteria. So there would be is a transfer of genetic material between two bacteria that are temporarily joined. Transduction occurs when fragments of the bacterial chromosome accidentally become packaged into viral progeny produced during a viral infection. So accidentally bacterial chromosome packaged into viral progeny introduced into another bacterium. By the viral vector. Transduction, okay. For bacteria. So transduction is like the meiosis, but binary fission is like the mitosis. So accidentally become packaged into viral progeny. It's transfer of genetic material between two bacteria that are temporarily joined. Now we have transduction. There only remains transfusion, which would be like, I think, the plasmid, right? So transduction. Fragments. So bacterial chromosome fragments. Bacterial chromosome fragments accidentally package into viral progeny, accidentally package into viral progeny during infection, viral infection, and then introduced into another bacterium by the viral vector. Introduced to another bacteria. Introduced to another bacterium via viral vector. So we have viral vector, we have bacterial chromosome fragments. For this one is sexual mating. Variability. Okay, we have question 13, which is chose D but it's A. A bacterial cell that does not rapidly cause a phenotypic change in the rest of the colony is likely not F+, meaning that this cell is not able to form a sex pilus for conjugation, making a correct expression of new phenotypic characters indicates that bacteria might have acquired genetic material from the environment through transformation or transduction, which occurs via bacteriophage infection. So let's see. We have, let's read the questions first. A bacterial cell is resistant to penicillin. The bacterium is transferred to a colony that lacks the fertility factor, so it's not conjugation. So bacterial genome transfer. Sterile. So lacks the fertility. Lacks fertility factor. So which of the following methods of bacteria is not likely to account for this change? So lacks fertility factor equals not conjugation. Conjugation, congratulations, you're married. Mnemonic. Let's add the mnemonic here. Congratulations, you're married. Conjugation. Congratulations, you're husband and wife. Your husband and wife, which is conjugation. Conjugation is sexual mating. Your husband, you may kiss the bacteria. Okay. Now we have and the rest of the colony does not become resistant to penicillin. So rest of the colony does not become resistant to penicillin. However, the penicillin-resistant cell has also started to exhibit or other phenotypic characters, including secretion of a novel protein. So let's go back to the explanation. A bacterial cell that does not rapidly cause a phenotypic change in the rest of the colony is likely not F+, meaning that this cell is not able to form a sex pilus for conjugation. The expression of new phenotypic characteristic indicates that this bacterium may have acquired genetic material from the environment through transformation or transduction, which occurs via bacteriophage infection. So transduction is bacteriophage, transformation from environment, DNA from environment, fresh from environment. So new phenotypic characters characters in case bacterium from bacteria got genetic material from environment, transformation, or conjugation or transduction. Transduction. Well, let's say that again. The options: transduction, infection with the bacteriophage. Or transduction, which occurs via bacteriophage, or transduction equals bacteriophage infection. So we also had here that transduction is bacterial chromosome fragments accidentally packaged into virus. So the whole bacteriophage thing is at play during transduction. So it loses bacteriophage transduction, and yeah, accidentally package into viral, introduced to another bacterium, viral vector. Duct vector, duct vector, it's a phage, bacteriophage. Okay. Let's even add it here to the mnemonic. Transduction, viral vector, bacteriophage. Okay. We got question 14 correct, which is prions are infectious proteins that cause misfolding of other proteins. Yes, prions generally cause a shift toward beta pleated sheets, so they turn alpha helixes to beta pleated sheets, causing decreased solubility and increased resistance to degradation. This ultimately leads to disease. This mechanism is very similar to the one described here for Alzheimer's disease, making C correct answer. And last but not least, we have question 15, which is a virus that requires, we chose B, but A is the answer. A virus that requires transport to the nucleus in order to produce viral proteins likely requires use of nuclear RNA polymerase in order to create mRNA that can be translated to protein. Therefore, only DNA viruses need to be transported to the nucleus to produce viral proteins. Eliminate all answer choices but A. Okay. Let's go to the question. After infection of a cell, viral particle must transport itself to the nucleus. What is the likely genomic content of the virus? So A is the answer. Double-stranded DNA, not double-stranded RNA. So let's look at the explanation again. A virus that requires transport to the nucleus in order to produce viral proteins likely requires use of nuclear RNA. Wait a second. A virus polymerase, RNA polymerase, nuclear RNA polymerase in order to create mRNA. Wait, so a virus that requires transport to the nucleus in order to produce viral proteins. So you want to produce viral proteins, you require RNA polymerase, nuclear RNA polymerase, to create mRNA that can be translated to proteins. So you need mRNAs for making those viral proteins. Therefore, only DNA viruses need to be transported to the nucleus to produce viral proteins. So why double-stranded, not single-stranded, is not explained here. Wait, yeah, negative sense, positive sense RNA. So extra questions. Cues. What are... So now we know what is conjugation. It's sexual mating for bacteria. What are sex pilus, you know. What are negative and positive sense RNA? And why, and this is from assessment Q15, why is a double-stranded DNA needed for after infection of a cell for a viral particle that must transport itself to the nucleus in order to produce viral proteins. Is it only proteins can be made only via double-stranded DNA? Why not double-stranded RNA? So the RNA is for making the protein. So yeah, it makes sense that you need the genetic material, which is the DNA that you need to make it from. Where does translation take place? Where does we know it's the ribosome coming in with the tRNAs, reading from the mRNA, right? But where does translation occur? We know cytoplasm of the cell. So translation is transcribed into mRNA. But transcription happens in the nucleus, right? Yes, you're correct. Transcription. Yeah. Okay. With the binding of RNA polymerase, an enzyme possible for RNA to a specific region of DNA called the promoter. Let's actually ask the question here. Viruses can use both double-stranded DNA and double-stranded RNA as their genetic. However, the choice of genetic depends on the DNA. Viruses such as herpesviruses, adenoviruses typically have double-stranded DNA. As when a DNA virus infects a whole cell, it delivers DNA. RNA viruses, on the other hand, can have either single-stranded RNA or for viruses like the influenza, the viral RNA is used directly as mRNA after infection. The host cell's machinery translates the viral RNA into proteins. But it has to happen in the cytoplasm. If you have to go to the nucleus, you need double-stranded DNA, right? For RNA viruses. So it can be only double-stranded DNA, or actually also single-stranded DNA to go to the nucleus for transcription. RNA cannot be used for transcription. The process of synthesizing RNA from a DNA template typically occurs in the nucleus. In the host cell transcription machinery leads the DNA template to synthesize RNA. The synthesized RNA, called messenger RNA, then exits the nucleus and enters the cytoplasm for translation into proteins. For DNA viruses, the viral genome is usually double-stranded. For DNA viruses, double-stranded is typically transcribed in the nucleus. For RNA viruses, single-stranded RNA can serve as mRNA directly in the cytoplasm. So these RNAs don't require transcription. We cannot transcribe. So RNA is not involved. It is involved because you are in transcription. You are making RNA from DNA. That's the whole point. So making. However, there's this reverse process that is kind of confusing me, where you make DNA from RNA. So, okay, is it possible to make DNA from RNA? It's like, reverse transcriptase. Yes, the process of synthesizing is known as reverse transcription. Reverse transcription is a characteristic feature of retroviruses, a family of RNA viruses, and it is also used in molecular biology techniques. In the context of retroviruses, HIV, reverse transcription is a crucial step in the replication cycle. These viruses carry an enzyme called reverse transcriptase, synthesizes a complementary DNA, cDNA, strand from the viral RNA genome. The resulting RNA-DNA hybrid is then used as a template to generate a complete double-stranded DNA copy. This DNA copy, known as provirus, integrates into the host cell's genome and serves as a template for the synthesis of new viral RNA. Reverse transcription is commonly used in techniques like reverse transcription polymerase chain, RT-PCR, to convert RNA into complementary DNA. RT-PCR allows scientists to study and analyze RNA molecules that is widely used in gene expression studies. While cells typically transcribe RNA to DNA during gene expressions, certain viruses and molecular biology techniques can utilize reverse transcription to generate DNA from RNA template. Perfect. Okay, let's now take a break and I'll be right back. We had to do the review for chapter 1. I don't regret it, to be honest. Okay, peace for now.